All right, welcome to our masterclass. This is all about getting you ready to absolutely ace the genetics section of your official life sciences exam. We're going to break down the big ideas, but more importantly, the skills you need to look at any document they throw at you and know exactly what to do. Okay, so let's jump right in. Look, the secret here isn't about memorizing a textbook. It's about training your brain to think like a scientist, to decode those complex exam documents and solve problems with total confidence. First things first, we have to lock down the one single concept that connects everything else in genetics, and that concept is genetic polymorphism. Honestly, it's the why behind every single question you're going to face. So what on earth is it? Well, putting it simply, polymorphism is just the genetic variety that makes us all unique. It means that within a population there are different versions, or alleles, of the same gene. Getting this is, without a doubt, the first step to understanding everything about inheritance and genetic diseases. And this picture? It shows you the whole process in a nutshell. Just follow along. You start with two parents, right? Each has their own unique set of genes. Meiosis creates the gametes, and then, and this is key, fertilization happens randomly. The result is a brand new generation of genetically diverse, or polymorphic, offspring. So now that we know the what and the why, let's get into the how. How is all this amazing variation actually created? We're going to look at the two main engines that drive this process, meiosis and mutation. You know, meiosis is so much cooler than just simple cell division. It is a masterclass in shuffling the genetic deck. You've got crossing over, where chromosomes literally swap parts of themselves. Then you have the random independent assortment of those chromosomes. It's a recipe for creating an almost infinite number of combinations from a single parent cell. Okay, so engine number 2 is mutation. Now think about it like this. If meiosis is shuffling the cards you already have, mutations are like adding brand new, never-before-seen cards to the deck. These are real changes to the DNA sequence, and they're the ultimate source of all new alleles and traits in a population. And this table breaks down the types of mutations you're going to see. A substitution, where one letter is swapped for another, might not do anything. But deletions and insertions, whoa, they're usually a much bigger deal. They cause what's called a frame shift. Imagine you shifted every letter in a sentence one space over. The whole thing would become gibberish. That's what happens to the protein. All right, enough theory. Let's put this into practice with a true exam classic, the pedigree chart. This is where you get to put on your detective hat and use your analysis skills to figure out how traits are inherited. But before you try to solve anything, you have to be fluent in the language of pedigrees. You just have to know these symbols cold. Square is male, circle is female, shaded in means they have the trait, and that line between them means they're a couple. It's non-negotiable. Okay, detective, here's your first set of clues. Look at the chart. Does the trait pop up in every single generation? It's probably dominant. But what if two totally unaffected parents have a kid who is affected? That is a dead giveaway for a recessive trait. Use these clues to make your first big deduction. Next up, you've got to figure out if this gene is on a regular chromosome or a sex chromosome. And the quickest trick in the book? Just count. Seriously. Compare how many males have the trait versus how many females. If you see it way more often in one sex, your alarm bells for sex-linked should be ringing loud and clear. Let's switch gears and head into the lab. Modern genetics is all about high-tech DNA tools, and you can bet your exam will ask you to look at the results of these tests and figure out what they mean, just like a scientist. RFLP is a big one. It's a key technique for spotting those genetic polymorphisms we talked about. Basically, you use these little molecular scissors called restriction enzymes to chop up DNA. Now, if someone has a mutation, the enzyme might not be able to cut, or it might cut somewhere new. This changes the lengths of the DNA pieces, and that's how we create a unique DNA fingerprint for someone. And this is what the result looks like. This is a classic exam document, so you have to know how to read it. Just remember this rule. The gel is like a racetrack for DNA. The smaller, lighter fragments are faster, so they travel farther down the gel. This creates that unique pattern of bands for each person's DNA. And you know, this stuff isn't just abstract science. These tools have incredibly powerful real-world applications. One of the biggest is in prenatal diagnosis, which lets us spot potential genetic diseases really early on. So let's do this. Let's analyze this right now, just like on the exam. We can see the DNA bands for the mom, the dad, and the fetus. See how the mom and dad both have a band for the normal allele, N, And the sickle cell allele, S, that makes them heterozygous. Now look at the fetus. Its bands line up perfectly with only the normal allele from both parents. So we can confidently say the fetus is homozygous normal, genotype NN, and will not have sickle cell anemia. See? You just did it. So we've covered a ton of ground. What's the master strategy here? How do you take all of this and turn it into a fantastic score on the exam? It all boils down to this winning formula, a 5-step process you can use for any question. 1. Always start with the core idea, polymorphism. 2. Figure out the mechanism at play. 3. Analyze the document like a hawk. 4. Combine what you see with what you know. And 5. Justify your conclusion with the evidence. That's the secret sauce, combining your biology knowledge with killer analysis skills. You're officially equipped. You've got the tools and you've got the strategy. Now it's your turn. Go find as many old exam problems as you can and practice this formula. If you do that, you'll be ready to walk in and master any genetics puzzle they put in front of you. You've got this.